Okay, folks, today we will be talking about enzyme kinetics with a brief introduction to kinetics. Our learning outcomes today, we will be able to list and describe the 6 classes of enzyme. We will understand some different aspects of binding substrate, and we will recognize some different types of cofactors that enzymes use. Today's Meet a Scientist moment is Maud Menton, MD, PhD. She was among the very first Canadian women to receive an MD back in 1912. She is most well known for co-authoring the paper describing enzyme kinetics that we will learn next time we are together. So what are enzymes? Enzymes are proteins that are catalysts, so proteins that do chemistry. So many proteins are catalysts, right? And they are absolutely necessary to our lives because enzymes behave as catalysts and lower the transition energy of most reactions happening in our body. Without these enzymes, most likely these reactions would not be happening. For example, these enzymes increase the rate of reaction by 10 to the 6th to 10 to the 12th. This is a massive increase in reaction rate. Additionally, since our bodies are relatively mild in terms of pH and temperature, this catalyst really helps speed things along when we're at almost a neutral pH, 7.4, and at a very low temperature of 37 Celsius. But enzymes are great because they are very, very specific at what they do. There is a new word that I'm going to introduce to you at this point, folks. Since we're talking about chemistry, we need to go back to some reaction schemes, but just make a note that in biochemistry, we do not call the things at the beginning the reactants, we call them substrates. Because why not learn more vocabulary? Thankfully, the product is still called the product. And as we'll learn in a little bit later, enzymes can be regulated. They can be controlled in a variety of ways, which is super important for our homeostasis. So how are enzymes classified? Well, a lot of times you'll see that the very end of an enzyme ends in ASE or A's. Now, in traditional naming, enzymes were named after their substrate. The enzyme whose substrate is urea, urease. The enzyme whose substrate is an alcohol, alcohol dehydrogenase, right? What do you think DNase has for a substrate? However, this type of naming is kind of challenging because sometimes we don't name the enzymes after their substrate. So catalase, for example, what is its substrate? It's a catalyst, which we already knew about enzymes, right? So we can break these enzymes into different classes to have a better understanding of what these enzymes do. And there are 6 major reaction types that will be good to know for biochemistry. And I think we'll see an example of each of these before the end of class. So the first type of enzyme class are the oxidoreductases. These are doing redox reactions. So they're oxidizing something or reducing something. Transferase is named super well also. It transfers a functional group to the substrate. Hydrolase performs hydrolysis, so it uses water to split a bond. A lyase performs an elimination reaction to form a double bond. An isomerase, you guessed it, isomerization. And then finally, a ligase forms a new bond, often requiring an input of energy through ATP. And to wrap this part up, folks, let's try to visualize this general reaction scheme for an enzyme. So in the very beginning, we have our enzyme and our substrate. First step is the enzyme binds to its substrate, giving us the enzyme substrate complex. Now, once enzyme binds its substrate, time for chemistry to happen. So we have a reaction performed, leaving us with the enzyme product complex. And then finally, the enzyme unbinds or releases its product. And this reaction scheme, folks, we're going to want to keep in mind over the next few lectures to kind of understand the various aspects we'll discuss. We'll discuss binding. We'll discuss chemistry separately, for example. Up next, binding.